Hello, welcome to our lecture on meiosis. We finished up talking about mitosis, which was cell division for repair, replacement, and cell or organismal growth. Now we're going to talk about cell division that is specific to the formation of gametes. We are diploid organisms. That means that we have two copies of each chromosome. But in order to maintain that chromosomal number, that 2n status, when we form gametes, our gametes must become haploid. And so two haploid gametes, which haploid is a single copy, those two haploid copies come together during fertilization in sexual reproduction and become a diploid organism. So we go from 2n to gametes that are 1n each, which will then come back together and give us a 2n organism. So sexual reproduction requires this fertilization event, and that's why we need our haploid gametes in order for this to create a diploid cell known as a zygote. Meiosis is the process by which we create these haploid gametes for sexual reproduction, and the formation of gametes is from diploid to haploid. Now, luckily, the process of meiosis very closely resembles that of mitosis. So like mitosis, meiosis is going to begin after a cell has progressed through the different parts of the cell cycle, G1, S, and G2. So it's moving into that M phase. But when it moves into M phase, instead of going through mitosis, it begins with mitosis, but then goes through a second cell division and moves into meiosis. So meiosis actually occurs in two stages. It occurs as meiosis 1, which is the initial cell division, giving us two daughter cells. And then those two daughter cells will divide again, and that's meiosis 2. So meiosis 1 is our first round of cell division, and meiosis 2 is our second round of cell division. But meiosis 1 does differ from mitosis in a couple of ways. First of all, our homologous chromosomes, right? So here we have two kind of white ones and two red ones. Our homologous chromosomes will replicate and form what are called tetrads. Okay, these tetrads are also referred to as bivalents. And what's going to happen is, I'll just kind of draw an X over here. So I have one chromosome that's been copied here. And if I change the color of my pen to this color, and then I have another one that occurs here. And this is dad's copy, and the other one is mom's copy. This changing pen colors is going to be tricky. And this will be mom. Now, when these two, if this is chromosome 1 and this is chromosome 1, right, I have a copy of mom's, a copy of dad's. I've replicated my genetic material, and now I have two copies here and two copies here. Before these divide, like we saw in mitosis, they're going to form a tetrad. So here's my mom's chromosomes there. And then I'm going to have dad's are going to form here. And so what happens is these guys meet. I have kind of this junction that occurs between them. And after this occurs, I'm just going to stick with these pictures down here now. When this tetrad or bivalent a reaction occurs here, it's known as a synaptonemal complex. And when that occurs, that synaptonemal complex occurs, these chromosomes are going to swap information. And this process is known as crossing over. So you can see here, these two guys got together and these two guys got together, right? And when they crossed, this red one took this section of the white one, and the white one swapped with the red one. So they swapped. So now what we have is we have chromosomes, right? We have chromosomes that are a combination of the original parents. So your gametes will have chromosomes that are a mixture of your parents' chromosomes. And this is sometimes why children are more closely related genetically to grandparents than they are to parents, because they have a combination of their grandparents' genes on the same chromosome. So this process is known as crossing over, and we'll look at this a little more in depth. So when a tetrad and bivalent is formed, remember we have these homologous chromosomes. This would be mom, this would be dad, and they come together, and right here, two of them are going to be touching, and this is where that synaptonemal complex is formed, right here, in between them. And where they start touching is where they're going to swap pieces so that we end up with a combination of the two chromosomes instead of one. So the process is called synapsis, and the actual structure that's formed that creates the bivalent is known as a synaptonemal complex. Now, this crossing over event contributes to genetic diversity. This is why we look somewhat like our parents, but not identical to our parents. So we had this contributes to genetic diversity to the human gene pool. It also only occurs in meiosis 1 during prophase. So immediately after interphase, when our genetic material has been copied and we're in that chromosome form, okay, so we've got our sets of chromosome, our sister chromatids. In prophase 1, this is where crossing over will occur. It does not occur at any other time in meiosis, only during prophase 1 in meiosis 1. So here's another look at that crossing over event. We have our homologous chromosomes condense up. They're really tight. We start forming the bivalent, and synapsis is going to begin. The bivalent is formed. Right, so here's that I'm sorry, it's going this way. So here's one, I was wondering, here's step 2. In step 2, our synaptonemal complex is forming, right? So here's our synapse. That's kind of what they're showing here. They're kind of gluing together. And then in step 3, you can see that these chromosomes are entirely attached to each other here in that synaptonemal complex. And then the event will occur. And these two here, they swapped regions of that chromosome. And then they start to separate back apart, and there are now referred to as a chiasma. A chiasma gets visible as the chromosomes start to separate from each other, and this occurs in very late prophase before we're going to move into prometaphase. Now, one thing to remember in this crossing over, genetic diversity, and only happens in prophase 1. Okay. Prophase 1. This process is also sometimes referred to as translocation. And we're going to look in the next lecture, we're going to look at the different forms of translocation. How much of this chromosome is being swapped? Are we swapping the same regions? Is it what we call reciprocal translocation or non-reciprocal? There are different types of crossing over that occur. We'll look into those details in the next lecture. How many crossovers actually occur in gamete formation is usually controlled by the cells themselves. So that's an important part of crossing over regulation.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
All right, so that's how we get crossing over and some
Crossing
genetic
over.
diversity in the crossing over events. So in meiosis 1, during prophase 1, we'll see that crossover event, the formation of bivalents after chromosome condensing. In prometaphase 1, which is not pictured here on the left here, but in prometaphase 1, the spindle apparatus becomes complete. Chromatids start attaching to those kinetochore microtubules, and the rest of what happens in metaphase, anaphase, and telophase 1 is identical to what happens in mitosis. Now, one thing I want to point out here is in metaphase 1 and anaphase 1. In metaphase 1, the bivalents organize along that metaphase plate, but they do it as a double row, like this. Okay? So they do this as a double row, and they randomly reassort. So this mechanism does not necessarily, this mechanism here, this random, now it makes sure, like, let's say that this is chromosome 1 and this is chromosome 2. It makes sure that there's one copy, one set of sister chromatids on one side of the metaphase plate and another set of sister chromatids on the other side of the metaphase plate. But which ones go where is completely random. So this is where we see Mendel's theory or law of random reassortment. This greatly contributes to genetic diversity. So maybe you got mom's chromosome 2, but you got dad's chromosome 1. And so this random rearrangement of these chromosomes along that metaphase plate in the middle there really strongly contributes to genetic diversity again. So we have two different factors in meiosis that really contribute to the genetic diversity of a species. That's crossing over and random reassortment in metaphase 1. In anaphase 1, we start seeing segregation of the homologs, and the connections between them break, and each joined pair of chromatids migrate from the center to the pole. So anaphase 1 here is just like anaphase in mitosis, same thing with telophase. And then eventually the cell will finish with, of course, cytokinesis. That's final cell division there. And these are just some pictures, actual microscopy-like pictures of this occurring in cells. Now in meiosis 2, it's a little bit different. In meiosis 2, we actually are going to go through mitosis. That's what's really happening. But there's no S phase. So in this case, I'm going to separate these sister chromatids again, but now the sister chromatids will be separated into a haploid state. I'm simply going through cell division again without DNA replication. So I don't replicate the chromosomes again. I just go straight into cell division again. So I go through meiosis 2, which is almost exactly like mitosis, but instead of ending with two haploid cells, two diploid cells in meiosis, I end with 4 haploid cells. And I now have the formation of the gametes, and you can see that they are different combinations, right? So this one's dark blue, light blue with a dark blue tip. This one's light blue and then dark blue with a light blue tip. So there's a different combination of alleles on each of these different sets of gametes. So this table is from your book. It is a really great way to compare mitosis, meiosis, the stages of meiosis 1 and meiosis 2. Meiosis 2, you can see no synapse. Very rarely is there any kind of crossing over during prophase. We don't want to say no. It's believed to occur once in a while, but it's extremely rare. They all pretty much have the same here, except for in meiosis 1 we see bivalents, whereas here we see sister chromatids. So sister chromatids look like this. Bivalents look like that. Right? So we have double the copies. They're stuck to each other. And then, of course, what type of separation occurs at anaphase. In meiosis 1, we separate the bivalents. In meiosis 2, we do the same thing that occurs in mitosis. So some of the things you want to think about in regards to the mechanism of meiosis. What is it? Why do we have meiosis? What cells would use meiosis? Take a look in your textbook and look at the different types of cells, names of cells that would actually utilize the process of meiosis. Would an epithelial cell utilize meiosis? Probably not. What is the purpose of it? Why do we have it? What is the process of crossing over? What happens during crossing over? What occurs during this event? What is a bivalent or a tetrad, and why are they important? What happens in a synaptonemal complex? What's occurring here? How does crossing over contribute to genetic diversity? Maybe think about what other parts of meiosis contribute to genetic diversity as well. What are the stages of meiosis and what happens in each of the phases within each stage? So this is meiosis 1 and 2, right? And then how does meiosis differ from mitosis? Some of the different phases are the same. Some of them are different. So you want to make sure that you're able to compare and contrast. So when you're asked questions on an exam, you know whether or not it's referring to meiosis or mitosis. So that's it on the lecture on meiosis. Meiosis is a pretty straightforward process. Make sure that you watch the videos on crossing over. And in our next lecture, we'll look at some of the chromosome structures. We'll get a little more in depth on crossing over genetic diversity and some chromosomal abnormalities. Have a great day.